This is a lecture on AP Biology Unit 1.3 through 1.6, Biomolecules. These next slides show, for each subtopic, what you must know and what you don't need to know. Use these slides to help you with your independent studying. You can see what you must know and don't need to know for 1.3, 1.4, 1.5, and 1.6. What's important to remember during this lecture is that structure dictates function. So as we assemble and disassemble these structures, these biomolecules, remember that they all carry out important functions in the body and that their structures govern how they function. A general term for what we'll be building and taking apart are called polymers and monomers. You can see in this picture that polymers are assembled from individual subunits called monomers. Here are two different polymers assembled from the same monomers. However, their arrangement is different. Amylopectin and amylose are both starches. They're both made from the same monomers, but the arrangement is different. This difference causes a difference in function. Monomers form polymers through a reaction called dehydration synthesis, also called condensation reaction. In order to form a polymer, you need to join bonds leading to the dehydration or condensation of water. So water is a product of monomers forming polymers. To break apart a polymer into its individual monomers, water must be added in what is called a hydrolysis reaction. For hydrolysis, think water, hydro, and lysis, breaking. The first biomolecule we'll examine are nucleic acids. Nucleic acids are polymers built from monomers called nucleotides. Looking more closely at a nucleotide monomer, it is important to know their general structure. In a nucleotide, you see three things: a phosphate group, a sugar, and a nitrogenous base. This nitrogenous base is adenine. These nucleotides can vary, and one way they can vary is their carbon sugar. DNA contains deoxyribose, and RNA contains ribose. You can see the difference between deoxyribose and ribose is the absence of oxygen in deoxyribose. To construct a polymer from two nucleotide monomers, you need to connect them via what's called a phosphodiester bond. Phosphodiester is a biochemical term where you see a double bond to oxygen, a single bond to oxygen, bonding to your carbon sugar on both sides, diester. A common theme you'll see as we build these polymers is the condensation or dehydration synthesis reaction. So to build a nucleic acid, you form phosphodiester bonds through the condensation reaction. Notice how water is a product and your two monomers are becoming a polymer. It's important to know the differences between DNA and RNA. I'll let you read these on your own and remember back to 9th grade biology. And we will cover this much more, especially its function, in our upcoming units. The next biomolecule is proteins. Proteins are derived from what's called polypeptides, which are the polymers. So when we talk about polymers and monomers, polypeptides are your polymers. The repeating monomers forming these polypeptides are called amino acids. And what's remarkable about proteins is that ultimately they are built from only 20 amino acids. 20 different amino acids. Remember how structure dictates function? That is definitely true in proteins because the order of these amino acids dictates how a polypeptide chain becomes an elaborate protein structure. The shape of these proteins is critical to their function. See, this protein, hemoglobin, is designed in a very specific way, and if something were to occur that breaks this structure or changes it, i.e. sickle cell anemia, this protein would be unable to carry out its function. Looking more closely at an amino acid, there are commonalities between all 20 of them. All 20 amino acids have the same general structure. We have a central carbon bonded to a hydrogen, an amino group, a carboxyl group, and what we call an R group. This R group is a general category for something more specific we'll see later. Here you can see different R groups. Looking closely at these amino acids, let's look for the amino group, the R group, and the carboxyl group. On every one of these amino acids, you can see the carboxyl group, the amino group, our hydrogen, and what's in white here are these different R groups. Another name for R groups are called amino acid side chains. To build polypeptides, you need to form a peptide bond between two amino acids. These peptide bonds occur between the carboxyl group of one amino acid and the amino group of another amino acid. Just like our other polymer formation, these polymers are also built by dehydration synthesis or condensation reactions. We saw this assembling nucleic acids. We're seeing it again for polypeptides. Two amino acids join together at the amino group and the carboxyl group, condensing water as a product and forming a peptide bond. Two monomers have connected to become one polymer. Protein structure is so important that we make sure we go into depth. This is an example of a polypeptide chain. You can see the repeating units of amino acids forming one long polypeptide polymer chain. This is not yet a protein, but rather a polypeptide chain. As we start to form a protein, this polypeptide chain starts forming hydrogen bonds. These hydrogen bonds start twisting and pleating amino acid subunits. So what was a polypeptide chain starts becoming either an alpha helix, you can see the spiral shape, or a beta pleated sheet. Look for these structures in protein models. You'll see them everywhere. What is governing these shapes? It's hydrogen bonding, which is covered in topics 1.1 and 1.2. From these alpha helixes and beta pleated sheets, other chemistry starts acting. The tertiary structure of a protein is dictated by chemical interactions between amino acids. Specifically, we look at the chemistry of the amino acid side chains. Going backward, we see that these amino acids, these 5, all contain electrically charged side chains. Aspartic acid and glutamic acid both contain a negative charge, whereas lysine, arginine, and histidine all contain positively charged, positive charges. You can bet that there's an attraction between an R group such as lysine and an R group such as aspartic acid. So let's go back to the tertiary structure, and we start to see some of these attractions. Here's an ionic bond, just like I predicted. You also see bonding such as hydrogen bonding, hydrophobic interactions, and disulfide bridges. We will go more into depth with this with a different lecture, but just know that the chemistry of an R group will ultimately dictate the tertiary structure of a protein. And now we can really see the protein start to take shape. Finally, the quaternary structure of a protein results when 2 or more different polypeptide chain form one macromolecule. For example, an enzyme such as DNA polymerase contains several different polypeptide chains working together to perform a common function. Here are examples of proteins. First of all, you see collagen is a fibrous protein containing of 3 polypeptides, coiled like a rope. That is a very different protein than hemoglobin, which is a globular protein consisting of 4 polypeptides, 2 alpha and 2 beta chains. So both of these are proteins with quaternary structures, different polypeptides working together. I won't go into depth with all of these, but understand that proteins are very important. They aid in enzymes, so carrying out bodily functions, facilitating chemical reactions, to be more specific. Defensive proteins, transport proteins, storage proteins, and others including receptor proteins, hormonal proteins, contractile proteins, and structural proteins. Notice how the wide variety of protein structure leads to a wide variety of protein function. I also want to mention, notice what's in the opening slides for proteins as you study for this unit. Just make sure you're studying what is in the scope of the unit. To break a protein apart, we call this denaturing or denaturation. So proteins can become denatured when these bonds that are forming the Quaternary, Tertiary, and secondary structure are broken. What can break these chemical bonds? Well, if you change the pH of the solution the proteins are in, or the environment these proteins are in, they can break. What can also break these chemical bonds, giving proteins their structure, are salt concentration, temperature, and other environmental factors. The next biomolecule are lipids. Lipids are the one class of large biological molecules that do not form polymers. The most biologically important lipids are fats, phospholipids, and steroids. The chemistry of lipids are nonpolar. This is hugely important because this nonpolarity leads to a hydrophobic interaction with water. The carbon-hydrogen bonds that are present in lipids are largely nonpolar bonds. So a polar water molecule will have very little interaction with them. So fats are built from two different structures. They are fatty acids, which are these long chains of hydrocarbons, and glycerol. Let's look at some more graphics of this. So in the gray here, you see the glycerol, and in the orange, you see the fatty acids. So to build these, we need ester bonds. And we saw these with building our nucleic acids. Notice the carbon double bonded to oxygen and single bonded to another oxygen. That's your ester linkage right here. That is a organic chemistry, biochemistry observation right there. Look at what we have. Again, the dehydration synthesis or condensation reaction. So glycerol connects to fatty acids, specifically 3 fatty acids, through 3 separate dehydration synthesis reactions, resulting in the product of 3 water molecules. and a final product called a triglyceride. Let's look at some different triglycerides and see how structure dictates function. Saturated fats have the maximum number of hydrogen atoms possible and no double bonds. So look at what that produces. A very flat, compact, stackable triglyceride. Notice how the picture here is of butter that at room temperature is very hard, very compact. Contrast that with something like olive oil, which is made from unsaturated fatty acids. Notice the double bond here, which will kink one of the fatty acid side chains, one of the fatty acids in this triglyceride. And if you were to try to stack more on top of one another, this double bond will really affect that. So it will become more difficult for these to compress and condense together, leading to a very different arrangement at room temperature. Notice the difference between solid butter and essentially liquid olive oil, all because of this double bond. Another very popular and very important fat are called phospholipids. So to go back, we are covering a few different kinds of lipids. We just looked at a triglyceride, and now we are looking at phospholipids. So going back, another example of a lipid is a phospholipid. And a phospholipid is very special because it contains both a polar region and a nonpolar region. So if you think about a phospholipid's interaction with water, it's very interesting. The polar head will interact with water and attract to water, whereas the nonpolar tail will repel water. We will cover this a lot more later because these are what make the membranes of our cells. Carbohydrates. Carbohydrates are a biomolecule, and they contain sugars and the polymers of sugars. The monomers of carbohydrates are called monosaccharides, and the polymers of carbohydrates are called polysaccharides. That's an easy one to remember, as long as you remember the word saccharides. You can see a monosaccharide here built into a polysaccharide. Monosaccharides generally follow a pattern. You'll usually see them in this ratio, 1 carbon to 2 hydrogens to 1 oxygen. Let's check that with glucose, which is C6H12O6. That's a 1 to 2 to 1 relationship. And glucose is the most common monosaccharide. 1 6-carbon sugar. Just like we've seen with our other biomolecules, monosaccharides combine to form polysaccharides through, again, this condensation reaction. We call this bond connecting two monosaccharides together as a glycosidic linkage. An example of a carbohydrate is starch, which is a storage polysaccharide of plants. Are made from alpha glucoses, functions of starch are energy storage. So you can see that they're stored in chloroplasts and other plastids. Glycogen, a storage polysaccharide in animals, is made from highly branched chains of glucose, alpha glucose. Glycogen is stored mainly in the liver and muscle cells. Same monomers, very different polymers. Cellulose is a major component of plant cell walls, which is a polymer of beta-glucose and is not digestible by animals. So there is a difference, albeit slight, between glycogen and cellulose. Animals cannot digest these long chains of beta-glucose that compose these plant cell walls. Notice starch, digestible by humans, and cellulose, non-digestible from humans. Very slight differences, but differences nonetheless. Another polysaccharide called chitin has a structural function found on the exoskeletons of arthropods and the cell walls of many fungi, like this grasshopper, with an exoskeleton made from chitin. One last important function or thing to note, and this is kind of thrown in at the end here, but I think it's a good preview. Just consider structure dictating function. When we build DNA, it has a very particular arrangement. We will get much more into this later, not really in the scope of this unit. But notice how the direction of these DNA backbones vary depending on orientation. But just understand from this slide is that the arrangement of monomers really affects the function of the polymer. So here we had some slightly different monomers. This is just arrangement. So that can affect the function of the polymer as well. Proteins have an amino end and a carboxyl end, which makes sense if you consider the polypeptide chain that builds a protein. Ultimately, you're going to have an end with an amino group and an end with a carboxyl group. So that just comes knowing the three parts of the monomers that build these these polypeptides. So if you know the structure of an amino acid, you'll know the end of a polypeptide chain. The types of bonds between monosaccharides determines the relative orientation in a carbohydrate. So you can see some very different overall structures. And we can divide this into primary and secondary structures of a carbohydrate. And these all have to do with the types of bonds between the monosaccharides. And this can have consequences. Consider what is digestible by humans and what is not. There's our theme. Change in structure equals change in function. So you can see hemoglobin ultimately taking this sickle shape. And that is solely due to just a switch in an amino acid here. And carry that through the secondary tertiary structure to the quaternary structure to improper functioning of those with sickle cell anemia. And if you want to, you can take this all the way back and think of, well, if these are the products of translation, then where did this mutation occur? Did it occur during translation, or did it occur maybe earlier in protein synthesis? These last slides are review slides. You can see amino acids. You can see nucleotides, the monomers building proteins, the monomers building nucleic acids. Sugar monomers building polysaccharides. This lecture did not cover disaccharides, which are just two, di meaning two monosaccharides bonded together. Sucrose, table sugar, and lactose, the sugar found in dairy products, are both disaccharides. And we see lipids here, three categories: steroids, phospholipids, and triglycerides. What was not covered in this lecture are the functions of steroids. They have a very important function in cell membranes, which will be covered later. And they're huge with signaling molecules, like hormones. That is the last slide of this presentation. This concludes mini lecture for 1.3 through 1.6.